Okay, so um, I'm Pavel Hrab from Ivano-Franko National University of Lviv, and I'm here to present um, roughly the only, and to my mind, the best app I created, named BGCViz. This is an application to visualize different biosynthetic gene clusters. Well, to begin with, I'd like um, to begin with what are those biosynthetic gene clusters. Those are gene clusters that are responsible for producing some kind of secondary metabolite. And actually, those metabolites, for example, antibiotics, are visually can be visualized because they color the media of the bacteria. But in recent years, those bacteria began sequencing, and several tools for annotating those gene clusters from the genomic data become available. The most used tool is called anti-smash, and from annotating the genome, it turned out that, for example, streptomyces bacteria have from 30 to 45 biosynthetic gene clusters, but only like 3 or 7 are active, and they produce those colorful media. And more than 20 biosynthetic gene clusters are cryptic or inactive. So the question is, what other of these inactive gene clusters are producing? And the problem statement for this project was, can we point areas of interest in the genome? So are there any clusters that are annotated with different tools? So anti-smash is kind of state-of-the-art tool, let's say so. But this task is not trivial. And therefore, there are many other tools that that produce slightly different results. The result of this project is a Shiny application, which for now integrates 4 inputs from 4 tools. This is anti-smash, Prism, DBGC. This tool uses deep learning for annotating biosynthetic gene clusters, and RRE Finder. This tool annotates RIPs, so actually peptides. The first 2 files were created by hand, so you need actually to fill in cluster field, start, stop of the regions, and a type of a region. But the data from DBGC and RF Finder can be uploaded right from the app. And you have a bunch of controls to filter your data. This app is mainly focused on filtering DBGC data because it have different scores, and you can apply different thresholds to visualize and explore it. And in the end, you can download filtered data frames, but actually it's only DBGC is filtered. So this app is not only for visualization of the data, but only for using an interface to visually filter the data and download the filtered one. The main task is to compare the DBGC data to the anti-smash one. And this is done by different approaches. The first one is to compare how many clusters are annotated with state-of-the-art approach, anti-smash, and how many of them are actually captured by DBGC. So actually here we're plotting an interception of those annotations. So the green one is only anti-smash annotated data. The pink or red one is both DBGC and anti-smash annotated data, so this interception. And the blue one is only DBGC annotated data. So this is new biosynthetic gene clusters, possibly new biosynthetic gene clusters. And from this data, I actually calculated rates, so annotation rate. So how many of the data is annotated by both tools, annullity rate, so how many of the many of the clusters are captured only by DBGC, and skip rate. So how many clusters DBGC missed from anti-smash data. And actually you can see that DBGC annotated a lot of clusters. And actually here I already filtered the data with domain threshold because many of the data is one domain, one protein cluster. It cannot be possible. So domain threshold set to 5, and a different activity score, which you can also choose from the select menu, thresholds, there are different results for novelty and novelty rate, annotation rate, and skip rate, so amount of clusters. So we can visually inspect which threshold you should use to get a little of new biosynthetic gene clusters that can be then identified by experimental tools. But not only the DBGC data can be compared. Actually, we as input we use also PRISM data, which is another tool for annotating gene clusters, and RRE finder data, which is for annotation for peptides. And here we actually plotting interception of data annotating by these tools using the threshold with the sliders with the reference. So reference is anti-smash from both RRE and DBGC, and for PRISM is DBGC and anti-smash. So you can see here which clusters are intercepting by those annotating by PRISM. For example, in the lower half of the chart. And chart is actually interactive, so you can see more data coming over hovering a mouse, a pointer. And the last part is all versus all comparison. So it's would be a silly idea just to compare DBGC to anti-smash because we have so many tools. And the idea of this comparison is to point at different and areas of interest, so clusters that are annotated by many tools. So if a cluster is annotated by many tools, it must be something. So this something was caged by different approaches. So here you have some circus map with links and a bar plot which tells us which cluster, how many times, with how many tools was annotated. Actually, here is a funny example because you can see one from DBGC cluster is annotated by 5 tools, but it only can be compared to 3 others. So to anti-smash, to PRISM, and to RRE. This is because I think this is a big, not cluster, but region. And this region intercepts with several clusters. That's why we have 5 time annotation, but the maximum we can talk about is 3. And yeah, check out the app. It is available at link below, and not now at GitHub, but will be available at GitHub soon. So do you have any questions? I would happy to answer them.
I have a question. So like what prompted you to develop a tool like this? What was the initial challenge that you were facing?
Oh yeah, so the initial challenge was there are many data and we don't know what to do with it. So actually the anti-smash itself gives a whole bunch of those clusters, and maybe they're not they cannot be experimentally validated. And different tools gives slightly different results. So the idea was, can we capture those slightly different results to point to some consensus between those tools? So are there clusters that actually are annotated by like a lot of tools, and those clusters can be then experimentally validated?
Understood. Okay. Thanks.
Yeah, thank you for the question.